In this video, we're going to talk about the protists. You'll find as we go through this video that a lot of the protists do not seem like they're very related to each other, and that's because they're not. The protist is a bit of catch-all group, and so these organisms can sometimes be a little hard to study because they're hard to organize together. Let's take a look. Keep in mind that protists are not a taxonomic group that is evolutionarily related, which is called a polyphyletic group. This makes studying them more complicated because there are no real similarities between the classifications of protists. Protists are more of a catch-all term for something that wasn't an animal, a plant, or a fungi. Thus, protists can fit into all of these other categories. The first type of organism we'll discuss are the algae. Algae are eukaryotic microbes that contain chloroplasts, so they can do photosynthesis and are thus classified as autotrophs, or self-feeders. There are two groupings of algae, true algae and secondary algae. True algae obtained their chloroplast by endosymbiosis of a cyanobacteria billion years ago. True algae can be single-cellular or multicellular. However, the multicellular algae have no tissue differentiation, which means, for example, plants have roots and leaves and stems, different types of tissues. Multicellular algae are just clumps of cells. Algae are found in a huge variety of aqueous environments, everything from freshwater to saltwater, or free-flowing rivers to standing ponds. They're also extremely important to life on this planet because they produce about 70% of the oxygen in our atmosphere as a byproduct of their photosynthesis. Interestingly, when prokaryotic life first evolved on this planet, the atmosphere had no oxygen in it. Oxygen didn't appear in the Earth's atmosphere until the evolution of cyanobacteria, the first photosynthetic organisms. At that time, they started pumping out oxygen as a waste product, leading to the death of 99% of the organisms on this planet, as oxygen's actually pretty toxic. There are two main types of true algae. The green algae, which contain chlorophyll similar to plants, and the red algae, which contain a different light-absorbing pigment that gives them a red color. Secondary algae are organisms that can do photosynthesis because they also contain chloroplasts, but the origin of those chloroplasts is different. Instead of ancient ancestors endocytosing a prokaryotic cyanobacteria, secondary algae were formed when their ancient ancestor engulfed a true algal instead, so a eukaryote that contained the chloroplasts already. This is an important difference because while true algae may rely on photosynthesis to survive, secondary algae can do photosynthesis and be autotrophs, but they also have the metabolism to allow them to use both organic molecules. Thus, they are both autotrophs and heterotrophs. The ability to have different metabolisms allows them to survive better because they can switch to whatever metabolic pathway is best given their environment. If they cannot get light exposure, they will not die, like a true algal would. There are three main classifications. Dinoflagellates and diatoms are considered microorganisms, while brown algae, or kelp, are macroscopic seaweeds. The dinoflagellates are mainly marine organisms and are an important component of plankton in marine ecosystems. They generally have two flagella that crisscross their body, which you can see here, one crossing through this way and the other crossing through this way, coming out the end. This often causes them to have a whirling motion. Some dinoflagellates are problematic at high numbers because they produce neurotoxins that can cause paralysis in humans or fish. Exposure can occur by contacting water that has the dinoflagellate toxins or by feeding on organisms that have eaten the dinoflagellates or incorporated the toxins directly. These are the organisms that cause damaging red tides that you often hear about. Other dinoflagellates are endosymbionts with coral that are vital to reef growth. These are what are being destroyed by rising ocean temperatures, leading to what's called coral bleaching. Diatoms, the other type of secondary algae microorganisms, are really pretty cool because they have an outer cell wall made of crystallized silica, which is why they make lots of different shapes when you see them under a light microscope, such as in this image. The fossilized remains of these silica cell walls are what's used to produce diatomaceous earth. While there are no algae that are direct pathogens, mostly because there's no sunlight in the human body so they do not survive real well to be a pathogen, they can still be medically problematic due to the toxin production we discussed with dinoflagellates. Often these toxins will get into humans through consumption of shellfish. Shellfish, like clams and mussels, are filter feeders, So if they filter water that contains the toxins become incorporated into the shellfish and are transferred to people after they are eaten. Different toxins have different physiological effects and are just named after what they do, as you can see from the list here. Another negative side effect of algal blooms, when there's too much algae in the water, which everyone has seen when the top of a lake gets coated in green slime, the algae eventually die and end up causing a drop in oxygen levels when they decay. The drop in oxygen lowers the oxygen percentage in the water, which can lead to fish kills that follow the algal blooms. The next type of eukaryotic microbe in the protist category are the amoebas. These are large predatory eukaryotic cells. They consume both bacteria and other eukaryotic microbes through phagocytosis, giving them a large role in the ecosystem. They eat smaller organisms and then in turn are eaten by the larger organisms. The defining feature of amoebas are the pseudopods, which they use for movement and also for engulfing food, like we will see in this video here. So here's the amoeba, here's a pseudopod reaching out and another one, and notice it engulfs this one paramecium. The pseudopods extend here, it's going to engulf that second paramecium. We fuse on the other side, and now both those parameciums have been trapped within this digestive vesicle. The amoeba are going to secrete digestive enzymes, which will destroy the cell membranes of the paramecium and digest those cells for nutrients. Most pseudopods are lobed, like we've seen in the video, but some have pseudopods that are more needle-like, such as this image shown here. These free-living organisms are found both in soil and water, and a few are known to be pathogenic. However, they don't really want to be in the human body. They only become pathogens when they accidentally end up in our body, which then becomes a new environment for them. The most common amoeba pathogen is Entamoeba histolytica. This forms a gastrointestinal illness that occurs when you drink water that has the amoeba in it. Another well-known amoebal pathogen that is very rare, but known mainly because of its catchy colloquial name, is the brain-eating amoeba. This organism ends up in the brain when you accidentally inhale contaminated water through the nose, and then the amoeba is able to make its way from the nose up to the brain. This inhalation typically occurs while swimming or diving in warm, freshwater places like ponds. Rarely, infections have been tied to tap water through the use of something like a neti pot, which is why you should always boil water before irrigating your sinuses with it. There have only been 34 infections in the past 10 years with this organism, 3 of which were caused by nasal irrigations. However, only 4 out of the 145 people known to have been infected since 1962 have survived the disease. Thus, while very few people get it, it is a very fatal disease. The third group of organisms we're going to look at are the alveolates. The defining feature that is similar between all alveolates is a series of flattened vacuoles called alveoli or sacs that lie just under the cell membrane. You can see them here in this image marked in blue, here and there. The 3 main groups of alveolates are the ciliates, the dinoflagellates, and the apicocomplexans. Ciliates are defined by their use of cilia for both movement and food acquisition. You'll see that here that your ciliates have what we call an oral cavity that is similar to a mouth. They use their cilia to beat food into this cavity where it gets trapped for digestion. The most familiar ciliate is the paramecium, which is the one that is shown in this image. You can see the cilia lining the outside. Paramecium are motile organisms that are often studied in biology laboratories by putting pond water under the microscope. Ciliates are generally found in aqueous environments, but they have no cell wall, so they need to be constantly pumping water out of the cell so that they don't burst. They do this using their contractile vacuole, just this center structure here. This vacuole fills with water from the cytoplasm and then contracts to push the water out of the cell. The second group that are the dinoflagellates we've already discussed when speaking of secondary algae. There are some dinoflagellates that do not have chloroplasts, thus are categorized as alveolates instead of algae. The last classification we are looking at are the apicocomplexans. This group contains some key human pathogens. Apicocomplexans are obligate parasites, which means they must spend at least part of their life cycle within another cell or host. This requirement means that they often have a complex life cycle, with certain stages of their development happening in one host and other stages happening in another. All organisms in this classification contain a structure called the apical complex that is found at one end. We can see the apical complex here with this series of secretory vesicles at this end of the organism. Apical complexes contain secretory vesicles and microtubules that allow the parasite to invade and enter host cells. For apical complexans that have multiple hosts, the definitive host is identified as the host where sexual reproduction, or fertilization to form the zygote, occurs, while the intermediate host is where asexual reproduction for proliferation of cell numbers happens. This group is of concern to us in this class because it contains a couple of key parasites, the first being the causative agent of malaria, which we'll examine on the next slide. A lesser-known pathogenic apical complexan is Toxoplasma gondii, which can be transmitted from cat feces, unwashed fruits and vegetables, or undercooked meat. Toxoplasma doesn't often affect adults, but it's one of the key pathogens that can cross the placenta as an associated with serious birth defects. This organism is actually why pregnant women are often told to use caution if they have to clean out litter boxes, because Toxoplasma can be passed to us from the cats. Malaria is caused by the apicocomplexan Plasmodium falciparum, which is spread from person to person through the bite of an infected mosquito. Thus, the parasite has part of its life cycle within the human host and part of it within the mosquito. There are about 228 million cases of malaria every year, with the majority of them happening in Africa and Southeast Asia. Of those cases, about 400,000 people die each year, 70% of whom are children under the age of 5. The malarial disease begins when an infected mosquito, here, bites an individual and injects sporozoites from the mosquito into the human cell. The sporozoites make their way through the bloodstream to the liver, where they first infect liver cells. Here, they mature to a form called a merozoite. The merozoites are then able to travel and infect our red blood cells, where they're going to replicate and divide, forming trophonoites along the way, until eventually those red blood cells rupture. This cycle continues repeatedly with infection and rupture of multiple red blood cells, often leading to anemia. Eventually, gametocytes are formed, or the gametes of the parasite. These gametes are going to be taken up by a different mosquito when a mosquito feeds on an infected person. Inside the mosquito, the gametes are able to develop until they eventually join together through fertilization and form a zygote. The zygote will form an oocyst, which will eventually burst, reforming the sporozoites, which travel to the salivary glands and start the process all over again. Based on this description of the life cycle, you should be able to identify that the mosquito is Plasmodium's definitive host because this is where fertilization and sexual reproduction occurs. The last categories of protists are the trypanosomes and the metamonads. These are mainly catch-all categories where they have additional modal eukaryotic microbes that have a flagella but are not dinoflagellates. You do not need to know a lot about these groups. I only mention them because there are two key pathogens. Trypanosoma bruceri, which causes African sleeping sickness, a disease with neurological symptoms such as confusion, tremors, and disruption of the sleep cycle, and Giardia lamblia, which causes giardiasis or beaver fever. Giardia is of interest because it's resistant to chlorine and sometimes can be a contaminant of standard drinking water. It's also one of the common causes of intestinal illness in campers and hikers, especially if they have drinking from mountain streams without purification.